For over 40 years, HIV has challenged humanity, reshaping lives and societies. The advent of antiretroviral therapy, or ART, turned HIV from a fatal diagnosis into a manageable condition, allowing millions to thrive. Yet, ART is not a cure. It's a daily reminder of the virus's persistence, as HIV hides dormant within our cells. The ultimate goal remains, a true cure that frees people from lifelong medication and fear of resurgence. Now, a new hope is emerging, not from new drugs, but from empowering our own immune system to control HIV. What if we could train our natural defenses to keep HIV in check, without daily pills? This shift in strategy could mean a future where the body itself maintains control, offering lasting freedom from the virus. We stand at a pivotal moment, with science and community driving us toward a new era in the fight against HIV. To understand the new hope, we must first grasp why ART isn't a cure. HIV targets CD4 T cells, the very cells meant to defend us, turning them into virus factories. ART halts viral replication, restoring health and preventing transmission, but it's not a final victory. Early in infection, HIV hides its genetic code inside long-lived immune cells, forming a latent reservoir. These dormant cells are invisible to both ART and the immune system, scattered deep within tissues. If ART is stopped, these hidden viruses awaken, reigniting infection within weeks. This is why millions remain tethered to daily treatment, facing lifelong costs and burdens. The latent reservoir is the core obstacle to a cure. To break free, we must find a way to eliminate or control these silent hidden viruses. Only then can we move beyond managing HIV to truly defeating it. The challenge is immense, but it's the key to ending the need for lifelong therapy. Understanding this is the first step toward a future without daily pills. The next step, harnessing the immune system to target what ART cannot reach. That's where the new frontier begins. The focus is shifting from attacking HIV directly to empowering our immune system. Instead of eradicating every trace of the virus, a functional cure aims for long-term control, keeping HIV so suppressed it causes no harm or transmission. The body would still harbor latent HIV, but immune cells would act as vigilant guards, preventing reactivation. This approach mirrors how we coexist with other persistent viruses like herpes without symptoms. The strategy: re-educate and boost killer T cells, reversing their exhaustion and training them to destroy HIV-infected cells. The goal is lasting immune memory, so the body is always ready to respond if HIV tries to re-emerge. It's a profound shift, from relying on drugs to strengthening our own defenses. If successful, it could mean a one-time treatment, freeing people from daily medication. This is about giving the body back control over HIV. The future of HIV care may lie within us. Scientists are targeting two key moments to train the immune system, right after infection, and in those already on long-term ART. Early intervention combines ART with immune-boosting therapies or vaccines, aiming to prevent HIV from establishing deep reservoirs and to build strong immune memory from the start. For those on ART, the kick-and-kill approach is used. First, drugs wake up dormant HIV-infected cells, kick, then immune therapies help killer T cells destroy them, kill. This all happens while RT continues, preventing new infections as the reservoir is exposed. The ultimate test comes with a supervised pause in ART, an analytical treatment interruption. Researchers watch to see if the trained immune system can keep HIV suppressed without daily drugs. Success means the virus stays controlled, or rebounds only slightly, without medication. These strategies, early intervention and kick and kill, are our best shot at preparing the body to face HIV alone. Each approach is tailored to the stage of infection and the state of the immune system. The goal: lasting control, not just temporary suppression. It's a careful, stepwise process, but it brings us closer to a functional cure. The immune system, properly trained, could be our strongest ally. Chronic HIV infection exhausts the immune system, causing T cells to express breaks called immune checkpoints. Viruses exploit these breaks, shutting down immune attacks. Enter checkpoint inhibitors, drugs that release these breaks, reactivating T cells. First used in cancer, they've shown promise in unleashing immune responses against tough targets. HIV researchers are now testing checkpoint inhibitors as part of the kick-and-kill strategy, hoping to wake up tired T cells to attack reactivated HIV. The approach is powerful, but not without risks. Overactive immunity can cause side effects. Researchers are working to balance effectiveness with safety, fine-tuning doses and combinations. Checkpoint inhibitors aren't a cure by themselves, but they're a potent tool for unlocking the immune system's full potential. They may be key to making immune-based cures a reality. Before human trials, new HIV therapies are tested in non-human primates infected with SIV, a close cousin of HIV. In groundbreaking studies, early immune-boosting treatments combined with ART allowed some monkeys to control the virus for years after stopping drugs. In chronic infection models, kick-and-kill strategies, using drugs to wake up the virus and checkpoint inhibitors to empower T cells, delayed or prevented viral rebound. These results show it's possible to train the immune system to control a persistent virus, even with a hidden reservoir. Primate studies provide crucial proof of concept and help identify risks before moving to human trials. While monkeys aren't humans, these experiments bridge the gap between lab ideas and real-world therapies. The successes are inspiring, fueling the next phase of research. Each breakthrough in primates brings us closer to a functional cure for people. Animal studies are the essential dress rehearsal for the main event, human trials. CRISPR, the gene-editing molecular scissors, has sparked hopes of cutting HIV out of infected cells. In lab dishes, CRISPR can snip out dormant HIV genes, hinting at a sterilizing cure. But delivering CRISPR to every hidden infected cell in the body is a massive challenge, and editing DNA carries risks of unintended effects. For now, CRISPR's most promising use is in the lab, modifying immune cells to resist HIV or fight it better, then returning them to the patient. This approach is safer and more controlled than editing every cell in the body. CRISPR may not be the main cure strategy yet, but it's a powerful supporting tool. It helps us understand HIV and could one day play a direct role in therapy. For now, it's an invaluable member of the research team. A functional cure for HIV would transform lives, especially in regions where daily art is hard to access. Lifelong medication strains healthcare systems and individuals, and stigma remains a barrier. A one-time or short-course therapy that trains the body to control HIV would be revolutionary. It would empower people, reduce stigma, and ease the burden on health systems worldwide. This mission drives scientists, advocates, and people living with HIV to work together. Clinical trials of immune-based therapies are now underway, applying lessons from animal studies to people. Progress will be gradual with setbacks and learning along the way. But the convergence of decades of research, new technologies, and global collaboration offers real hope. The dream of controlling HIV with our own immune system is no longer distant. Science and community are building a future where daily pills are no longer needed. The journey is personal, urgent, and filled with optimism. The next chapter in the fight against HIV is being written now. Together we're moving